Hello guys, so I'm creating this video just to give you a kind of capsule for the practical guide for the gene cloning, and then it will be kind of very easy for you to understand all the next step. So we will start it from the identification of a gene of interest. So whatever gene you want to clone, you have to identify it using different database. If you want to clone the yeast gene, you simply can go for Saccharomyces genome database. You can also get the sequences from the NCBI and etc. And then you have to choose which vector you want to use. I mean, where you want to express your gene of interest, and based on that you have to choose the vector. For example, for bacteria you can use PET21 series vectors. For yeast you can use PS vector or Pichia pink for Pichia pastoris. For insect cells you can use baculovirus vector, and for human cell line you can use pcDNA or Trex series vectors. And then you will perform a PCR amplification of your gene of interest, which will contain the flanking region for the restriction enzymes. So you remember that if this is your if this is your DNA, and then your primers will annule in this way, and then your reverse primer will annule this way, and these are the flanking regions which has the restriction enzyme digestion site. Then purification of the PCR product using agarose gel electrophoresis column purification. So you, after doing the PCR amplification, you will run agarose gel electrophoresis, and then you will identify some big blobs like this on the gel. You will cut them out, and then you will use the Qiagen gel extraction kit to purify the PCR amplified product from these bands. And PCR amplified product from these bands. So this is a very purified PCR product. And then the next step will be the restriction, digestion, and ligation of the vector and gene of interest. So what you will do, you will take a vector. For example, you will take PET21 and gene of interest, and you will digest them with NHE1 and XH1, both of them, so that you can create the flanking region for the both of the restriction enzyme. And you remember how I created. So for example, it's like, so this is your vector DNA, and after restriction enzyme digestion it will look something like this. So these are the flanking regions on both the sides, and then the same is applies for the vector DNA. And then you will again do the gel purification. So for example, here you will do a gel purification. Let's just take this out. For example, you will run the agarose gel electrophoresis, and maybe you will find two bands, which one will be the high molecular weight, and one will be a very low molecular weight, right? For the vector DNA, and you need the larger one because this is the main fragment in which you will ligate the gel purified restriction and digested gene of interest. So you will take out this from the gel, and you will follow the gel purification protocol. Similar kind of digestion you will do for the gene of interest, and again you will purify the gene of interest from the gel. Then you will ligate the reaction mixture and put it for 22 degree for 30 degree C, and after this ligated mixture you will use this mixture for the bacterial transformation. That's how you will only select whatever will ligate in this process, and those colonies which will have which will take in this ligated plasmid PCR product, and they will grow on the antibiotic resistance plate, because only after ligation a bacterial plasmid can become a supercoil. A linear fragment cannot be supercoiled, and that's why it's very hard for a bacterial cell to internalize a linear DNA fragment rather than a circular and supercoiled DNA. So the bacteria, after ligation, it will be a complete circular DNA, and then it will be easily transformed bacterial cell. Then you will identify the right construct against antibiotics on the LB plate. So those who will get this PET21 vector will acquire the ampicillin resistance, and only those cells can grow on the ampicillin plates. Then what you will do, you will take out, for example, if this is your plate and you get colonies like this, a lot of colonies, so you will probably pick these colonies. For example, you will pick this one, you will pick this one, you will take 4 colonies. Sorry, I think I have to use this way. Yeah, just to give you some examples. So you will pick these 4 colonies from the plate. You will take out these 4 colonies from the plate, and you will do the plasmid. You will purify the plasmid using Qiagen plasmid extraction kit, and then you will verify the correct construct. There are 2 ways to do verification. There is one called primary verification, which is using the restriction and digestion. So you will take these colonies, you will grow them in LB medium, which should also contain the ampicillin drug, and only those cells will grow who will have this plasmid. And then you will perform a mini prep. The mini prep means you will take roughly 5 ml of bacterial culture, and from that you will purify the plasmid DNA. And this plasmid DNA, that means the ligated plasmid DNA, which you just ligated here. Okay, and then you will use again this NHE1 and XHO1 enzymes. So you'll take 4 plasmid, you will add the NH1 XH1 enzyme, set up the restriction digestion reaction, and after that whatever product will come, you will again run agarose gel electrophoresis, yes, in 1% gel, and then what you will identify here is that, for example, if there are 4 constructs, in 2 of them you will find, in 3 of them you will find only some restricted products. So for example, out of these 4, only these 3 which has after restriction digestion will give you 2 fragments. One is for the vector and another is for your gene of interest, and the 4th one might be just negative, just a false negative result, false positive result. I'm sorry, it's just false positive results. So now you will only concentrate on these 3. You will take these 3 DNA, and you will final verification you will do using DNA sequencing. And maybe after DNA sequencing you will get to know that this is the one which is the correct construct for you. So this one is confirmed. So the clone 2 is the correct construct, and then you will prepare the glycerol stock of this clone, and then you can store it for forever for further experiments. Yeah, so this is just kind of short sort of step-by-step guidance for how the cloning thing should work for any kind of gene of interest to clone in any vector based on the restriction enzyme digestion cloning. Yes, thank you.